Action potential arrives at synaptic knob. Calcium channels in membrane open, allowing calcium to move in cell. Increased calcium causes synaptic vesicles filled with acetylcholine to fuse with membrane. Acetylcholine is then released from synaptic knob by exocytosis into the synaptic cleft. Acetylcholine is picked up by receptors on motor end plate of skeletal muscle. Sodium-potassium channels open, allowing sodium ions to enter muscle fiber. Increase in positive ions inside muscle cell initiates an action potential on sarcolemma. Potential travels down sarcolemma into T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calsequestrin, into the sarcoplasm of the muscle cell. Calcium then floods over the thin filament. Inside the thin filament, calcium binds to troponin, pulling tropomyosin off the active site on the actin. High-energy myosin heads leap to bind on the exposed active sites on the thin filaments, forming a cross bridge. The myosin head pivots to the low-energy state, releasing ADP and moving the thin filaments over the thick filaments by way of the power stroke. ATP binds to the myosin head, causing the head to break free from the active site on the actin. The myosin head becomes energized as ATP is converted to ADP. Calcium is pumped back into the sarcoplasmic reticulum and attaches to calsequestrin, allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle.
Fascicles. The fascicle is a bundle of muscle cells or fibers. Inside, myofilaments are made up of thick and thin filaments. Narrow plate-shaped regions of dense protein material called Z-discs separate one sarcomere from the next. The darker middle part of the sarcomere is the A-band, which extends the entire length of the thick filament. The I-band is a lighter, less dense area that contains the rest of the thin filaments, but no thick filaments. A narrow H-zone in the center of each A-band contains thick but not thin filaments. The sarcolemma is the cell brain of the muscle cell. It is surrounded by mitochondria in the sarcoplasm.